My name is Haridas. I'm with the Leica Microsystems, and I'm here with actually two of my colleagues. You can see Louise, Bertrand, and James Marr. They are at the system. So I will just briefly introduce what we are going to do, and they will be the one doing remotely showing what it is. Right? And so the goal here actually, so let me start my short presentation. So what I'm going to present to you is called CORAL Cryo workflow. This is a Leica solution for your cryo-electron tomography. So it's a light microscopy solution before taking it to lamella milling or even TEM imaging what you're doing, right? And so in your typical electron microscopy TEM lamella workflow, you have taking sample to plunge freezing to then before getting into the EM side, in between the sample quality inspection or identifying the right structures of interest, that is where we come in, this particular aspect. Right? Like has other whole suite of solution for sample prep, but what we are presenting today is primarily focused on this aspect, cryo-light microscopy, so that you can identify the structure of interest before taking it into the EM system. And why do we need that? So typical pain point, okay. You know, electron microscopy time is expensive, and if you have like a large grid and to identify a small area of interest quickly, right? Yeah, please.
Yeah, that's your first goal.
So is my sample worth it? Did I prepare a good sample? That's identifying that. And then also finding your sample target area quickly. Then specific target. If you are looking to do DEM at a very specific area, how do you identify first, right? So basically the fluorescence imaging many times actually helps you to get there quickly. That is where the cryo-light microscopy comes in. So unlike typical microscope, regular microscope, you are imaging it under one liquid nitrogen temperature at under cryo condition. That's the goal. And for that, to that end, actually, we from Leica we have two solutions. One is a wide field solution we call Thunder Imager EM Cryo-CLEM. So, and another one a true confocal solution, STELLARIS 5. STELLARIS is our confocal platform. Thunder is our wide field imaging platform. Plus, actually, it also has deconvolution and computational clearing to remove out of focus signal. And to get a one level higher resolution, you can get to the next level of confocal. So the workflow here all involves you using fluorescence imaging and as well as often fiducial marker beads embedded into the cells or around the tissue, right? So that you can 3D localize the area you want to mill or image in electron microscope. So taking it to confocal 3D imaging or even thunder imaging, then taking it to electron microscope SEM and identifying the areas, then finally doing the lamella milling, then taking it to the transmission electron microscope. This is the whole workflow, right? Because you need an ultra thin section for transmission electron microscope. That is the goal of lamella milling here. And this is the one solution is this wide field solution. It's a camera based solution, but also, as I said, this has a deconvolution option plus a computational clearing, which is actually allowing it out of focus signal removal. More importantly, there is a transfer shuttle to bring in the sample at under cryo condition without devitrification to the sample stage, or to take it back into a closed or electron microscope system, right? And also there's a dedicated objective, which is a cryo 50x.9 MA objective, which is one of the highest MA objective for cryo imaging. And the cartridges can take auto grid or bare grid options. So second option is a true confocal system, a point scanning confocal system based on our STELLARIS platform. This again, both shares this transfer shuttle and cartridges objective. This infrastructure is shared between them. similar solution. Then why do you go to confocal? Because again, if you want to localize in 3D, confocal has higher Z resolution as well as a little bit higher XY resolution as well. Right? So you can precisely locate the area of interest. And on the confocal side, it is actually, there are some unique capabilities on the confocal side, which is actually based on a spectral confocal. That means up to 5 spectral detectors. That means actually you don't need a filter. You can position the spectral window wherever you want. Means any combination fluorophores you are using, right? You don't need to worry about it, which one you are choosing, right? And then additionally, some capability like acoustic optical beam splitter is again without dichroics, electronically tunable splitter. Then more importantly, and one option is an optional item, but very crucial item, white light laser for excitation, which allows you to excitation laser also position wherever you want. When you combine the spectral tunability and white light laser together, you have complete freedom of permutation and combination of fluorophores. So this again, then on top of that, confocal also can be, the resolution can be enhanced by deconvolution. So our Leica's proprietary solution is called lightning. So if you add up lightning on top of confocal, you can further enhance the resolution in the confocal image, right? So full spectral flexibility, then additionally lightning deconvolution. Then this further to take it to the electron microscope. Next step, what you need is localization, identifying the area of interest for imaging, and then mark it, coordinate it, and send it to another EM system, right? For correlation purpose. So through that, and also we have a new workflow which is actually built on. If you have a 3D confocal imaging, this is actually 2D image, and this lateral one, these two, what you are seeing is cross section, XZ cross section or YZ cross section, Z direction, right? Vertical, you can see membranes and inside intracellular part and the other bottom membrane, and then you can mark the position. And we have also built in a interpolation system which will allow you to mark the coordinate at a higher than what optical resolution will allow you. So you can position the marking even at a much higher resolution. So that then second part capability is that many times actually in this workflow, we need to know the vitrified ice's thickness. Is it the right thickness, or how far is it getting crystalline contamination, or you are getting devitrified ice? So for that also reflection imaging can help you. Because if you here in grayscale image, what you see is a reflection imaging. And you can use that to identify, measure the thickness of ice. And then further enhance it further. One more level capability is that this laser is actually a pulse laser, and our detectors are photon counting detectors. This means you can make use of fluorescence lifetime, not just the intensity, lifetime. Why do we need? Typically, biophysicists used to use this technique, use a pulse laser, then do a curve fitting, and then color code the lifetime into a color coded map. But what we have done is, to make it easier, we have completely taken it a step, completely eliminated the curve fitting part, and created a whole bunch of tool set called tau sense tool set, which is actually tau contrast, tau gating, tau suppression. These are all lifetime based, but eliminates the need for your curve fitting capability, right? So if you do that, what you can achieve is reflection removal. For example, this is a grid reflection removed by time gating, because reflected light comes instantaneously, whereas fluorescence has a delay. You separate it, instantaneously you can separate it. So you don't need to necessarily discard it. You can actually create another image of reflection and fluorescence into 2 channels simultaneously, or multiple fluorophore separation. You can do that, right? You build upon that, and then the whole workflow where actually you create the stack, mark the position, lamella, identify the lamella. So if you have fiduciary markers, identify it and export it as a coordinate system so that you can correlate with the electron microscopy. So I'm not going to get into more details here. It's better to watch how it is done. So I'm going to transfer it now to Louise. She's at our Thunder EM system, and she's going to take over and present that workflow. Okay? And at the end, we will have a 10 minutes for question and answer. So, Louise.
Thank you, Haridas. Let me know if you can see my screen.
Not yet.
Yes?
Yep, you can.
Perfect. So here we have the Thunder EM Cryo-CLEM. This is a wide field system, camera-based, filter cube, and cryo stage with cryo objective. We use what we call the CLEM software that is based on matrix screener. It is a workflow, so we start from the first step all the way to step number 6. We define our experiment. In this case, I will just go back here. I load my experiments that I acquired earlier. It's a 2 grid, so we can select either the auto grid or the bare grid, like Haridas mentioned. We select the CLEM and we have 300 mesh. We create the experiment. I already created the experiment because we are limited on time. I have 2 jobs here. The first one with the arrow is called the autofocus job. That will allow me to find the best focus plane on my grid at different positions. This one is based on contrast. 60 micron range is usually enough. We can go higher, we can go to 100, and you have the choice to decide between speed or accuracy. And I will show that to you in a few minutes. Usually I will start with bright field. I can find easier my grid, and I will go to our structure of interest that is in fluorescence. Then we have a 5 channel. So the sample today are provided by Jay, this right lab. There are HeLa cells that are grown on, I believe, gold mesh. They are auto-grid. In green, you will have the METO tracker. In yellow, I have autofluorescence of beads at 1 micron. In magenta, they are the 40 nanometer, and in blue is the ERCH. So let's see how it looks when we go through the workflow. The first step is to do an autofocus. So I will move to the middle of my grid. I will zoom in, and now you can see that I'm already in the middle of my grid here. I can say, let's do an autofocus in the white field. And once the autofocus is done, it's pretty good. You can see the mesh here, you can see the part grid here. If you are exporting into, so we will be using marker to select the grid alignment as well as the structure of interest. If you want to export into serial EM, it is very important to align the template with your grid. So we will do the second step, that it's a small spiral. We'll do 1. And I will do the grid alignment. If you go to the Aquilus, if you use the map software from Thermo Fisher, you don't need to do a grid alignment. This is not important at that moment. So I will turn on my tools to do my grid alignment. I will decrease my image opacity so I can see behind how the grid is located. As you can see, J inserted the grid. It's pretty well aligned. This is my center of the grid, and if I bring the opacity to 0, this is the center of the template. And I will just now do a small rotation. What is important is to have the main axis perfectly aligned, meaning in X and Y, maybe a small adjustment in one of the other direction. If I bring my image back to opacity here and I zoom out a little bit, I can see I have a small orientation problem. And maybe moving a little bit in X, or not X, but Y, and now in X. Oops, sorry. And that looks pretty good. So what will happen when we export? We will have in the top quadrant plus X plus Y, in the bottom quadrant plus X minus Y, etc. When this is done, I want to know where are my cells. So now I will redo a spiral, but this time I will select my fluorophore, that is the green, and I can do the full grid. So I can say, let's do a spiral of 4 rotation from the center toward the exterior. And again, now you can see where the cells are located. You can also notice that the grid is not flat, because here, if I look toward the middle, my cell seems in focus, but as we are going toward the periphery, they are not in focus. So I will not go through the whole grid. We'll select a certain area. We'll go to the next step. Once we define the area where we want to acquire, the next step will be to create a focus map. So I will select one of my tools here to select the area that I want to image. I can say that I want to image maybe a small area. We'll do a 2x3. I can put some focus point. I can do each one of them, or I can do every second one. Then the software will interpolate between the area where there is no focus point. From there, it will use what we predefined before. So our capture through 60 micron, 9-step, and I will start my focus map. If I want to see how the focus is, whoops, sorry, wrong channel. Let me stop that 2 seconds. Let's select the appropriate channel and let's redo our focus. Now you can see it finds the focus nicely. So it will do it for 4 positions that I selected. And if we are happy with the focus map, we can keep going with the next step, that is run matrix. Or we can always bring back our focus that we just defined, go live, double click on one of them, and then when I look here, I can decide either I readjust my focus manually and I set it back, or I accept it as it is. In this case, we will be doing a small Z-stack, so that's pretty good. So next step, I want to do a Z-stack. If I go to my job where I will be acquiring my 5 channel, I will be right now probably in the focus position at this level. The Z-stack will be defined according to my focus point. Let's say we'll do 8 microns. If I go live again and I go to the beginning and the end of my Z-stack, it's giving me 24 sections. We'll go back to start. We'll turn on our matrix, and I will click Run Matrix. And now you will hear probably the filter cube switching. I will show you here the acquisition of each image. In the meantime, I pre-selected OME, that is the Leica pyramidal structure to export all the images. You can also select TIFF, JPEG, etc. The mosaic will be merged using a smooth algorithm. I combine all the channel, I combine the mosaic, so I have one image, and at the end it will apply an extended depth of field. What it means, it will be taking the best focus on each section, bringing that together as a 2D image that will be your reference image if you want to look before you export the file into maps or the coordinate into serial EM. So now it might look a little bit overexposed. I didn't apply an auto range here, that is auto scale. I can apply the auto scale for each of the channel. If I'm not interested to look at the bright field, I can just turn it off, and then I can see my cells. Of course, every time I start a Z-stack, it goes back to the beginning of the Z-stack and go all the way down. So my image shows up in focus for a certain period of time, and then after that, out of focus as it's going through the Z-stack. Few more tile here. So we're using an STMOS camera, so it's a 19mm field of view. It's good resolution, and it's a very sensitive camera. So we can see here in yellow that are the 1 micron beads in aqua in the Texas red. They are more autofluorescence. You can see on the bar grid the autofluorescence that we can detect with the wide field. Couple more tile, and then we will set our marker, we will export our data, and I will show you how we pass from raw data to small volume computational clearing. That is part of our Thunder imaging. So the small volume computational clearing will reassign the light where it's supposed to go and remove the out of focus if there is still after the deconvolution. And I will turn off this window since we are almost at the end. Okay, so once we have our mosaic that is aqua, and we can do it for the whole tile, we need to use our marker. The first one will be for your grid alignment. Usually we find area that we can identify into the Aquilus or into the cryo-TEM. In this case, I took a small area, so I will say where the mesh are broken, it's a good place. Maybe this area, if it's not covered of ice, and just for the purpose, I will select a third point. The second one is our cells of interest, or our structure of interest. So if I turn off the transmitted light, I can say I have a nice cell maybe here. If I move in the z-axis, that's in the middle of the mesh. We'll just select that one for the purpose of the demonstration, and maybe this one here. The third one is for your fine alignment. So if I zoom in, and now I turn on maybe my green, my yellow, I can see that I have the 40 nanometer beads inside the cell. So I can mark a couple of them. They are much more than 40 here, so I will select the small one. And I will do that for few cells. So if I look here, I have again few of them if I move in the Z, maybe a couple here, and we'll select some outside as well. So I can keep going, but again, we're a little bit limited in time. So, whoops, delete this one. Next step will be to export our coordinate. So I will go to my export file. Move that to the middle. And when I'm under the export file, I have few possibilities. I can export with maps from Thermo Fisher, so it can be imported directly to the Aquilus, or I can export with the latest version of serial EM, or I can go with an old version if your cryo-TM use an old version of serial EM. The serial EM setting, the scale here is not perfect, but Dr. Yan published a paper that is called Correlator Interactive Software for Real-Time High-Pressure Cryo-Correlative Light and Electron Microscopy, where you can use it and you will get perfect correlation. This is our camera that is selected here, and I have the folder where I want everything to be saved. And I will click Export Data File. As I export, I will have my screenshot with where are my area that I identified, so I can find them in the Aquilus. And I will also have my mosaic combined with everything on it. If I go to serial EM, Then at that point, I just need to select serial EM here and do the same thing, export data file. Then I will have, for my grid alignment, the position that will be identified with a minus or a plus position in XY. We do not have the Z position here. Once everything is done, I can now go outside our CLEM software. I can see, for example, a good type, maybe this one is good. I will go to my process and I will say I want to do a small volume computational clearing. So if I select again this tile with a nice cell in the middle, I will go to my process tool, I will go to lightning, small volume computational clearing. The refractive index is very important because we are doing a deconvolution. If I'm using vitrified, a sample in vitrified state, the refractive index is 1.31. If you are like me and sometimes you don't want to change it, you can use 1.33, that it's like water. The first channel will not be processed, it's bright field. The other channel will be processed. We can tweak the parameter if we want. Regarding the thunder imager EM, the parameters are read directly from the microscope, so you don't need to touch that, and you click apply. As we click apply, it will go through the process. Oops. Might have some bandwidth issue. Uh-oh, looks like my software decided not to work. I will transfer to James, and I will show you at the end the difference between them. So James, your turn.
All right, sounds good. It's always fun doing a live demo. I'll make sure to leave you a few minutes at the end. Can everyone see my screen?
Yes.
Okay, and hopefully can hear me all right because you answered. So I'm going to go through the CORAL Cryo on the confocal. So instead of having a wide field instrument, we now have an instrument with a confocal scanner. You can see here that our interface looks quite similar to what Louise just showed us. I'll skip most of what she's already shown us and highlight more of the confocal rather than the wide field functionality. So for our workflow, we have a similar cascade. So we start with a file manager where we can reload previous settings. We do still work in a job-based fashion, so we'll have jobs where we go through and use those for our workflow. We have the grid, either bare or auto grids. We have an experiment manager where we can set up something automatic as well. We do have a cryo pump manager now in this software. So for systems sold with the cryo pump that is a little black box, you'll get a temperature readout of your stage, the ambient temperature, the pump speed, and your liquid nitrogen level. There's actually a little warning voice as well if your temperature starts to drop, so you always have that nice audio feedback, knowing if maybe you have a blockage in the pump. We'll minimize a few of these as we go along. So we still have Spiral Scan Manager. You see I have a bunch of jobs set up. So at this point, I'm actually going to switch over to our jobs to give you an idea of what I have set up and show you a little bit about the confocal. So mine's a little bit more complicated because I wanted to show you that you could set up multiple different jobs. I still have a wide field camera, so we can do 3D or 2D imaging in wide field, just like we did on the Thunder CLEM. I do have one set up for doing a spiral, and then I have two jobs for doing confocal. And I have this confocal example, so I can walk you through a little bit of the features on the confocal instrument itself. So first I want to highlight that we are a spectrally based detection. So each one of these lines below here are detectors. There are no physical barriers, so there is no notch filter or bandpass filter that is in front of these detectors. It is a mirror-based, prism-based detection where I can move these and actually change where they're at in the spectrum. If I double-click this, you can see that this one is from 500 nanometers to 546 nanometers, just short of this line, and it's actually my reflection channel. So I'm doing a reflection measurement to measure the ice. Above that is a white light laser. So this line here is indicating the spectrum in which I can choose laser lines from. So for the STELLARIS 5, that is from 485 nanometers all the way to 685 nanometers. And I can dynamically change any of my laser lines within that. So if this one is set to 545, I could do 546, 547, anywhere within there. This is for the STELLARIS 5. We also have the STELLARIS 8 that has the expanded white light laser that goes from 440 nanometers all the way up to 790 nanometers. So you can really hit that near-IR excitation as well. So we have spectral-based detection, a white light laser, and then as Haridas mentioned, we have that pulsed ability of our white light laser. So there's a few additional functionalities I want to show you. Here we have an image. So I have a transmitted light on just to show me the grid bars. And then this is a 545 excitation trying to collect around Texas red emission. So if I go live now, you can see that I have this cell structure. And actually in this channel, what I'm looking for are these bead-like structures. So these should be roughly 1 micron beads like we saw on Louise's sample. And I did forget to mention this is the same sample type as Louise. So everything here is just a mimic of that. But you can see in this channel, I have a lot of autofluorescence potentially from my cells. So I want to see if I can split that based on my tau separation. So to do that, it's simply going to tau modes, selecting from the drop-down, tau separation. When I select tau separation, an additional window comes up where I can set in time where I try to split my 2 channels. I've already set this up. I won't go too deep into how to actually set it up. But essentially what I can do is now I'll have 2 channels. So sorry, that's my reflection. I want to look at this one, and we'll bring it down. We'll focus a little bit. So you can see that I'm actually splitting off something where I can split one channel versus another and see if I could separate those 2 signals. I did have this set up earlier, but I think it's shifted slightly, so we'll continue forward. So I can, oh, I did it on the wrong detector. I apologize. So I meant to be on my actual Texas red detector. This one I had set up previously. Obviously for a reflection, everything would be instantaneous, so I wouldn't really see too much difference. There we go. Now I have this autofluorescence from my cells, and I have what is the grid bar, but also the autofluorescence from the beam. So I could separate those 2 signals quite nicely. The other mode talked about was tau gating. So for this sample, it's a little harder to show. So what I've effectively done is forced a little bit of reflection in this channel. So if I go live here, what I should see is my cells, or sorry, the 40 nanometer beads, and I'm getting reflection off my grid bar. If I were to go to tau gating, now I get an actual time window where I can split my signal based on the arrival time from the detector and the fluorescence. And you can see I've completely removed or mostly removed that grid bar. Let's say it's something that's actually of interest. So maybe you don't want to throw away the signal, you want to keep it. If I open that window back up, I can actually add a second channel where I do the opposite time window. And now I can have grid bar versus my fluorescence. So the tau intensity or the tau modes give you a lot of functionality to use lifetime-based measurements to either divide signals, get additional information from the overlapping fluorophores, or remove unwanted signals. So there's a lot of functionality you have within that. So we have multiple settings here. So this is our example. We still have confocal settings. We can do a Z-stack, all sorts of things. So now we'll move back into the experiment, and I'll actually show you how to do the exact same thing Louise did, but all with a confocal image. So back inside the experiment, I have my grid. I could position myself around that. I could move physically my field of view. Over here, we have our spiral. So I'm going to use my wide field GFP to do just a quick 2 spirals near the center. And I will quickly make sure that I'm in focus. So this button up here will drag me to my current field of view. I'll try not to zoom in and out too much just to not make anyone motion sick in the room. There we go. So we're nicely in focus, and I'll click spiral down here. So what we're really going for is we're setting up multiple jobs to allow us to work within the workflow, and then as we go through the workflow, we use those in specific locations. So this is a very nice grid. It's very flat, so I have a lot of area to choose from. To keep this within a manageable time, I'll just do a simple 2x2. So again, with the draw tools that Louise showed, I'll create a 2x2 pattern. We'll set up the same focus map, but I'll do every single one. So I can either do that manually, or I can have it set it up by a defined interval. So I'll just do an interval of 1. We could go to interval of 2, 3, or do other specific intervals. We have autofocus jobs that we can use, like Louise showed as well, but here I'm actually going to use my confocal job, just because I can do it faster by hand when we only have 4. So I'll open my focus map, we'll come to this first location, and I'll go live. I could turn channels off and on, so I have multiple channels on currently, and you can kind of see the power that you get with the confocal. Since it's prism-based detection, multiple light lasers, multiple detectors, I can actually have multiple channels running simultaneously. So whereas Louise had 5 channels that ran separately, I actually have 3 channels currently that are running all together, and I can see those simultaneously. All right, so I have my focus map set up. I then go to my overview scan manager. This is where I select what this overview will actually be collected as. For this one, I'm going to use my 2-dimensional confocal scan. I'll apply that to it. You see a little highlight name come up telling you that that's the job that's going to run for that overview. And then the next step in my process is to click overview. It'll then go to each one of those tiles and take every confocal image setting that I had put up. So that's going to be a reflection, a transmitted light image, an autofluorescence of 1 micron beads, mitotracker green on mitochondria, 40 nanometer beads, and a Daffy channel. So I'm collecting many, many images, 7 in total. So as soon as that finishes, I could do things like disable channels. Let's say I only wanted to look at mitotracker green to really understand where I want to take my lamella. You can see I could turn many things off of here. If I zoom in a little bit, I can also hide. I'm going to make my interface slightly smaller. I'll hide that region so we can see that a little more clearly. So now we have an overview of the confocal GFP, sorry, mitotracker green, and I can start selecting areas that I want to take lamella from. I can even bring back my crossbars or my reflection to get an idea of where they're at inside the grid bars as well. So at this point, you can exit just like you would on the Thunder CLEM and take this overview to say a CORAL-EM if you only want to know where things are placed. This workflow though is targeting going to lamella going to the mill and actually fib milling lamella locations to do TEM. So our next step is to actually target lamella locations. So we have 2 panels here. One is the scan manager. This is the job I'll use for the lamella imaging and localization. And then I have the targeting. So I have alignment marks, lamella locations, and feed locations for doing the high resolution 3D targeting. So for this one, I didn't take a very large area. You would have taken a larger overview to give yourself a really good idea of where you're at on the grid. But I do have the center of the grid, so maybe I'll mark a location there. This one's actually quite intact. I usually look for broken grid squares, but maybe we'll just choose a couple of what look like thicker ice or things that would be unique to the SEM image. I then mark lamella locations. So I'll choose some cells that are maybe towards the center. We have this one that's by itself. We have these two down here that might be of interest, so I'll choose the one on the lower portion. And then just to round out to 3, we'll choose one here. So after I've chosen those 3 locations, I can assign a job that will actually go there and take our 3-dimensional confocal image. So I'm going to use my confocal scanner that is 3-dimensional, assign that job, and again, we get that indicator of where the field of view will actually be collected. I can then actually review the lamella that I've chosen and see where each one is located. And then I can mark beads. So if I were to bring back either my magenta channel or my yellow channel, which is the beads, the 1 micron beads that sit outside the cells, I can choose a few locations of each one of those. So in the essence of time, I'll only choose a few, but you can mark as many as you would need to get a proper 3-dimensional correlation to the EM. So once I've marked some bead locations, my lamella locations, and I'm assigned a job, my final step to collect the data is to click lamella. It'll now go, it will ask me for a job name. So we'll keep this one as project and just call it like a demo. It also gives you a unique identifier. So each day you use the instrument, every grid you use, you'll have a unique identifier that gives you information about the day it was taken. So if you're cataloging these samples as you go along, you'll always know or have a reference of what was taken and when. So at the top here, it's collecting the data. Again, we're going through those 7 channels in 4 different settings and taking a Z-stack. As that runs, I'll point out a few more things of just our workflow. The final step will be doing the save EM marker file. This is unlike the Thunder CLEM, it just exports all of the 3-dimensional data. You can also choose additional file types like a screenshot, saving the list, some other formats and things like that. While we're in this window, there's many other things you can manage. These are usually kind of set it and forget it settings, but if you want to look at your cryo pump, change your carrier settings, and another very useful one is under the cryo pump, once it's connected, you can have that audio where it tells you if you're below temperature. All right, so we're on the second one here. Each one of these takes roughly a minute. I do want to point out, you can't quite see the system, but we do have a sensor on this, our new stage that actually raises and lowers the objective for putting samples onto the stage so you don't bump your objective, and then you don't have to manually raise the objective to do that anymore. What's really funny is I think I'm in a fishbowl. There's someone looking at the window right now. So the final step in this is doing the 3D target. So I've marked lamella locations, I've marked beads, but right now it doesn't actually know a Z coordinate for that bead location within my Z stack. So the most important piece of this workflow is setting that 3D localization so that you know where and how to target the lamella so you get the most precise milling possible. And I'll show you one quickly like that, and then my final step will just be show some example data that we took earlier so you could see what lightning looks like as well as the ice measurement from a sample. So as soon as this ungrays, we'll know we're done. I'm going to bring up our side window so we can see what's going on currently. We're on that final channel. So these are the 7 channels that I've collected. You can turn them on and off on the right side. So for lamella targeting, I could maybe only keep on my mito tracker green. for my three-dimensional bead targeting, I could keep on maybe my magenta, which is the Texas red, and the 40 nanometer beads in yellow. So I'll select my first lamella. It'll bring up the XY coordinates that I marked for the beads and the lamella. And then what we're actually going to do, so I'll make this a little simpler before we get too far. There we go. So if I zoom in here, what we've really strived to do is, if I am pixel-based, I'll turn off smooth rendering, you can see each step in my Z-stack is a physical pixel. If I were to not have interpolation, when I move this point down to a target, I can't target it more precisely than the pixel. But if I interpolate the motion between those pixels, I can actually get a more representative or high resolution targeting in Z based on the intensity differences through that pixel stack, and get a much better localization in Z to give myself better targeting when I go to lamella middle. So there's the actual lamella location target. The other ones are the beads. So if we come back up here and I drag this bead down, you can look for, it should be towards the bottom. Oh, I have the wrong one on, sorry. I need to turn these two on, because I'm actually targeting this one. There we go. You can see that I can precisely put it right over the top of that one. So once I've marked all these locations, I can export my data. That final step would be exporting the marker file. We'll zoom in because I have that screenshot enabled, because I do want to have this reference image showing me where I put my markers. And we can go and actually look at the data that would have been exported. So you can see it gives us an experiment, a day and time, and then all the images. We also get the coordinate files out that Louise showed. And then to finish up here, I want to give it back to her so she can also finish up what she was saying. I'll show you some lightning data. So this is on the left side, raw data, and on the right side, that deconvolved high resolution image. If I zoom in, I'll try to go a little slowly because I know that things sometimes go a little slower across. You can see that my non-deconvolved is quite hazy, a little blurry, but you still have the structures there. And once I deconvolve it, it gets much more clear. We get better resolution, and we can actually see that we have better Z resolution as well. So you can do much more precise targeting with this lightning data. If I turn off the magenta, or sorry, the mito tracker green, you can also see those 40 nanometer beads, probably clustered, but you can see them much more clearly. Turn the yellow off as well. One last piece of information I wanted to show was actual ice measurement. So if I zoom out a little bit here and modify my lookup table so I can see this a little more clearly, and we'll do clipping through 3D. We can very nicely see our ice, it's a reflection measurement, and our signal within that. So you could measure this thickness to get a good estimate of how good your plunge freezing is, or how potentially bad your ice is, or how thick the sample is just in general. With that, I think hopefully I left a few more minutes for Louise. And if you want to take back over, I'll give it to you now so you can show the end result of the summary.
Thank you, James. I will take 2 seconds, not even. So on the left side is the raw data, on the right side is the deconvolved data. I will remove the white field or transmitted light. I will do like James, I will zoom it. And I want you to see that when we have raw data without deconvolution, we have all the scattered light, we have all the out of focus, we cannot even see the 40 nanometer beads inside the cells, whereas after the convolution, we get much higher contrast, little bit better resolution, higher contrast because we remove the out of focus. And after that, if you want to get much higher resolution, then we go to the STELLARIS Cryo. And on that, I think we will open for question, correct?
Yep, that's correct. If you have any questions, feel free. James or Louise or myself can I try to answer your questions?
Do you have to empirically determine the laser power as to not devitrify, or is that automated somehow?
It's empirical, but at the same time, the experience with the white light laser is that we don't see any devitrification from the laser power. Typical what is needed for imaging, you can, of course, increase the laser power significantly and force it to do it. But because of sensitivity, these are photon counting detectors with high sensitivity. So you tend to use very low laser power, relatively low laser power in.
So I guess I'll ask you sort of a stupid question, but in the workflow, where and how do you actually move the sample now into the cryo-electron microscope? You just sort of physically walk it across the room, or what happens next?
So that part actually is we didn't, so we were focusing on our the microscope side only, but this transfer shuttle we showed here. Let me go back to
I mean, I saw your picture, but
this one, right? So this is designed so that you can bring the sample from a grid plunger or something and load it in the cryo condition, and then image it, then take it to your electron microscope under cryo condition, right?
so that same holder is exactly the same for the.
Yes, you can, that cartridge, you can pull it out into the transfer shuttle or push it into under the microscope, right? And then take it to the EM using that same transfer shuttle. That's the idea.
So you walk it from one room to
Yeah.
the other.
So ideally, like probably Jay can comment about more because we use it right here. So we have the Aquilos system sitting next to the same room with the confocal system, right? Similar, that's the ideal situation, but even if it is in another room, this protects the sample from vitrification, devitrification.
So
Like what Haridas said, if you're going to move from phlegm to the alcohols, you can reuse that cartridge because there is a Leica cartridge from the TFS, and then it's compatible with this cartridge. And then if you're going to TEM, you have to upload the grid and then put it into the autoloader
Oh, wow.
and then put it into
yeah,
the
that's what I was wondering.
yeah. So if you go to TEM, yeah, you have to like unload
Okay.
it. Yes.
Individual grids.
Yeah, individual grid, but it's durable.
It's doing
Yeah, it's totally durable, right?
The grids have to be clipped for this.
Prior to this, or?
Yeah. But I think bare grids also can be used, right? Not necessarily it has to be clipped, right?
Yes, yeah, so you can use, so this one, we are clipped grids already, because the clipped grid have more capabilities. And then so that you image here, and then when you load it onto the TEM, basically your grid is barely changed. So that's why, but you can definitely image bare grid first, and then clip it. But just during the clipping, you may damage the grid. So the image that you had before may be gone.
Do you typically clip then most of the time before you use the cryo?
Yeah, so we do both, but right now I clip the grids.
I would like to know something about whether this procedure and instrumentation is available here and whether this can be used by anyone or
It's right here next
your
to.
collaborator or something.
That's actually, Jay might be the right person because you have it right here.
Yeah, it is over there. This belongs to the national facility, which is what they talk about, like the National Cryo Center. So you submitted a proposal, and the proposal is approved, which is a very high chance to get approval, and you can use it.
I see, and then can you do it hands-on, and do the students get trained or?
Yes, the students will get trained because the proposal includes a training portion. Yeah, so the students will get trained by the staff and they use it. Yeah.
And the proposal will include both the fluorescence imaging and the cryo-EM.
So the proposal should be, so I think it's like, the project managers from our facility would be a better one to answer those questions. But yes, so the proposal would be a scientific proposal, like you have the scientific questions that you want to ask, and then you need those tools to answer it. And then we'll have a panel of like external experts will, judge your proposal, and then if it's approved, and you can use the system to solve your questions. Yeah, but you cannot just like, I want to use the system.
If the proposal is approved, do you have to pay for the data collection? Or
No,
is it-
if it's approved, then it's NIH funded project. So you don't have to pay for it. Besides your lottery.
Is the FIM part of that as well?
Yes,
Okay.
it's also part of it. So the TM, and then the CLEM, all of them, it's supported by the National Center. Yes.
Any more questions?
Yeah, is the link on your website how to apply?
Yes, yeah, the link, if you go to the UW-Madison Cryo-EM Research Center, you should see a few centers and go to the MSEC, which is the Midwest National Cryo-EM Center, and then there is a very large buttons that you can click, and then this is like submit a proposal, and it's very straightforward, yeah.
Any other questions?
It's kind of related to the earlier question about the intensity. Is there no risk of bleaching, having all these fluorescents going?
So the one thing actually typical, at least the current wisdom is that under cryo condition, right, there is a reduced bleaching. Probably because that oxygen transfer and everything is reduced, vitrified through vitrified ice. And that's the general consensus. That is what is kind of significant. Some people claim no bleaching, but I don't know whether that is accurate, but it's significantly less.
Thank you. The transfer shuttle, does it have, it hold also the liquid nitrogen
Yes.
inside the
Yes,
shuttle?
there is a kind of a reservoir underneath it. That is how you are keeping it protected. And the gas vapor for actually kind of a positive pressure so that humidity won't get in.
Okay, yep.
Yeah.
Something
Yeah.
workshop, which is 4 o'clock, it's already 4. So I'll be going to the for the second one.
Thank you very much.
Thank you very much.